I am Aritra Bose. I'm a research scientist at IBM Research. And today I'm going to talk about some methods we have developed based on topological data analysis and higher order statistics and applied it to understand some epidemiological questions related to severe COVID-19 and its relationship with metabolic syndromes, hyperlipidemia, and so on. So I think you guys have already seen and known about COVID-19 crashing the metabolic party, where COVID-19's the SARS-CoV-2 viruses, it enters through the ACE2 receptor in our system. And the ACE2 connects with the renin-aldosterone angiotensin system, which is in short called RAAS. And RAS has a profound effect in our, in regulating a bunch of things related to our metabolic activity and cardiovascular system. So when ACE2 enters through that, it disrupts a lot of that, causing many downstream kind of effects and causing a lot of comorbid diseases, along with just severe like COVID-19 and make it more severe. So we wanted to investigate this a little bit more, and we were looking at certain literature where we found very conflicting kind of results related to how hyperlipidemia relates to COVID-19 and even for the severity of COVID-19. So we found that there are certain publications that refer to dyslipidemia and hyperlipidemia to increase the risk of severe COVID-19 and they also some people also found that they were insignificant when in trying to relate to severe COVID-19, and then there are also papers showing that they are protective and they are actually negatively related to severe COVID-19. So with that, we were scratching our heads, and we wanted to kind of again like understand this problem now a little bit better, and we kind of surmised that can we leverage massive clinical records data so to kind of understand this question better. And also another relevant question would be that what role does RAS play in kind of exacerbating COVID-19 to make it more severe and in some cases fatal? And also, how does the clinical variables, because metabolic syndrome, which is kind of triggered by RAS, they are actually like a host of diseases together, right? So they have very well like defined kind of literature in comorbid diseases, and and as the COVID-19's the SARS-CoV-2 virus disrupts all of that, it would be good to understand how these variables interact with each other and then lead to the explanation or elucidate how RAS impacts the system. So these were our motivating kind of points to do this work, and we've also found that like we kind of thought of developing new data-driven approaches to understand these epidemiological questions better, which are beyond just logistic regression based association studies. With that, we kind of probed the Explorers data, which is one of the largest clinical records databases in the US, and it has about 56 million records. And we filtered for missingness, and we randomly selected 1 million records from Explorers, and just to kind of establish a random cohort. And then we also kind of obtained all the COVID-19 incident patients from Explorers, which resulted in about like 270K patients. And then we also selected like equal number of controls to have a matched C19 cohort. And these are all the variables that we studied that included disease incidence. This also had like the drugs that were administered to these patients, and also their demographic variables, including the the racial and the ethnic categories. And Explorers uses the CDC defined variables, so we just went with that. And there can be a discussion differently that how CDC's definition can be improved, but we just went with what Explorers kind of had it. So then obviously we did some like basic epidemiological kind of studies with the logistic regression approach, and we found that okay, hypertension is strongly associated with COVID-19, and that was very well known, so we are in the right direction. Then we asked this question that whether the RAS drugs as defined by the ACE inhibitors and the angiotensin receptor blockers or ARBs can they, when they interact with hypertension, can they be protective of severity of COVID-19? And we do see that it is protective in here. And then, but then the hypertension was kind of, more positively associated with a very high OR, odds ratio. So then we asked the same question that we started from: how does hyperlipidemia fare? And we found that hyperlipidemia, again, it's very conflicting across different cohorts. When, for example, in the African American cohort, hyperlipidemia was negatively associated with COVID-19. And for example, in the in the random cohort, it was positively associated, and then in severe COVID-19 cohort, it was insignificant. So we are back to square one. We are still scratching our heads. So then we kind of surmise that this is probably like just the logistic regression is not being able to mine this, and we need better and sophisticated tools to kind of ask these epidemiological questions. So then we kind of thought of cumulants, and we what we call as redescriptions from the cumulants to form and answer these epidemiological questions. So if you have not heard of cumulants before, it is a higher order statistic. It kind of captures the higher dimensional interactions between different variables in your data, and it can be a multi-omic data, it can be clinical data, any data. So any variable, that sense, which is a random variable, and you can take that, and cumulants is defined as the log of the moment generating function. So the first order cumulant is essentially the mean, the second order cumulant is the variance, and the third and fourth order, so on and so forth. When they are standardized, they're known as skewness, kurtosis, etc. So we now we had this cumulants method, and we want to, and there's a very good property of cumulants that when you do higher order cumulants, the lower order cumulants, if they are independent of each other, they cancel out the joint cumulants. So you have a true way of seeing what is the signal with the n way or k way higher order interaction between these variables, right? So now we are talking about grouping this variable together, so which are known as redescription groups, and it is analogous to something which we kind of in epidemiology very well known as endophenotypes, where endophenotypes have like the similar genetic liability in the background when you group them together, these phenotypes. So we did the same, and we computed cumulants on this data. We then obtained the Jaccard distances between these different redescription groups. And interestingly, we see and this is completely unsupervised, by the way, right? And we see a cluster just related to severe COVID-19, which we didn't, play with. So the data told us that there exists a cluster, and so that was very intriguing. By just looking at the Jaccard distances between different cumulants, that there exists this very nice block in the middle of severe COVID-19, right? So with that, we wanted to probe that more further. So then we turned onto another very similar higher dimensional algorithm or technique, which kind of talks about the shape of the data, which is from topological data analysis. And we wanted to extract these topological features from the data, which are kind of important. So just some quick primer. So we sometimes call this as a simplicial complex, but it's very simple to understand actually, and it's simplicial in that sense as well. So you have like this point space where you have like these interacting points, and then the simplicial complex essentially defines the finite collection of sets. So you see all the possible points here grouped together, and then we also introduce something called filtered simplicial complex. And why we do that is essentially, again, is very easy to understand that the subcomplexes are part of the bigger complex, and as you kind of you can define a monotonic kind of non-decreasing function, and you can get those subcomplexes from a bigger complex. So with that quick primer, we can go from data points to barcodes, right? And these barcodes are essentially the features of the data that are getting captured. So every shape of the data you can think of like naively getting transported to some barcode, and then the barcodes that live longer in this space. So for example, this in this graphic again, sorry. So Yeah, here you could see that as we move the barcodes, right, they kind of die off as well. So that kind of talks about importance of those shapes in the data. So there's one long barcode, which is the entire point cloud, right? So for example, in here, in the second figure, you can see that in the second figure, when we filled the first kind of hole in the data with three interacting points, the second homology group, or H1, is kind of like, triggered, right? So you see the birth of the bar there. And then in the fifth figure, if you see that there is a hole in it with four interacting points, so you have like the first birth of the bar in the third homology group. So with that, we applied the TDA on the same cumulants data, and we wanted to do this persistent homology kind of bars for each of that, and we obtained about 1,000 cycles in that. And these cycles, the top leading cycles, were from the data. And this is very interesting. Again, this is entirely data driven, so we have not tweaked anything per se to obtain, right? So this is just the data talking to us with TDA. The main cycles were sex and RAAS, and then followed by calcium channel blockers and chronic kidney disease and RAAS, followed by obesity and RAAS, and then interestingly followed by African Americans and RAAS. So we didn't know anything about African Americans being induced or kind of treated, or having a different like perspective of severe COVID-19 just looking at the data, except that they were, they had high odds ratio. But here the data is telling us that the African American cohort per se has to be studied in a, differently, with like also in more detail, with probably some genetic liability that they have towards severe COVID-19 and metabolic syndrome related disorders, which the data is kind of telling us, right? And that is also attributed in previous literature. It is very, over the years, because this was still evolving when COVID happened, but now we have a better understanding, and there is a lot of papers followed that indeed looks into like African Americans and try to understand why they had like more liability of the disease than others. There can be environmental conditions and other things as well in this, right? So, but also we found that hyperlipidemia is all across these cycles from in the data. So that also answers our first question that we started asking, which logistic regression kind of failed to come up with, that how does hyperlipidemia fare? And we see that hyperlipidemia is very important when we are trying to study severe COVID-19, unlike what was seen before. So essentially the takeaway is that logistic regression can be...
Oh, okay, cool. Okay. So I resume, right?
Okay. So yeah, I mean, we are going back to the question of that we were trying to answer with hyperlipidemia and severe COVID-19. And now it is the not so curious case, right? Because when we probed, we saw the odds ratio to change significantly for, the with the cohort for COVID-19 with and without hyperlipidemia in it. So it shows that hyperlipidemia does have an effect. So then we moved on to for another analysis based on cumulants and redescription groups, which we call as CUNA, the cumulant-based network analysis. And in here, we take the same redescription groups, which are in the higher order, and we embed that into a graph with the variables being the nodes, and then the edges between those variables being the strength of the connection, the number of times they appear together in higher order in these redescription groups. So the width of in this network of the edges are related to how strong those connections are between certain variables. And we did also perform some network centrality analysis on the same network, and also perform community detection. And we found the top 5 nodes that are most important in this network are actually RAAS, followed by hyperlipidemia, followed by beta blockers, then African Americans, and then followed by severe COVID-19. So this kind of like ties our story together, right? Because we started probing hyperlipidemia, we found that African African American cohort were treated differently if in the data, and then that showed in our results in topological data analysis. And then with this further analysis in CUNA, we see them appearing again as one of the most important factors in the in treating severe COVID-19 and related metabolic syndromes. So this is kind of like the complete story, right? And we also found that the most important and the strongest interactions were between RAAS and severe COVID-19. So again, it points at everything points to the direction that RAAS drugs, as well as the related metabolic syndrome drugs, such as angiotensin receptors blockers or ACE2, can be used. So ACE inhibitors can be used to treat severe COVID-19 related metabolic syndromes. And also, we have to kind of treat the cohort for African Americans a little bit in a more detail to understand the genetic basis of such kind of difference in treatment or in susceptibility of COVID-19. So our key findings is summarizing here. I already did some. That RAS is a key driver in mediating severe COVID-19 and related metabolic syndromes, which is not a new news. I mean, but we did find from the data again and again that this is important. Hyperlipidemia is strongly associated, and it has far-reaching implications in severe COVID-19 treatment. And African Americans appear to be one of the most important variables in CUNA and in TDA, which was not present in logistic regression analysis. So this is very important to understand that epidemiological questions that we ask in our data with these large, massive data sets mostly restrict ourselves to doing log odds ratio and try to understand like different associations. They may be limited in scope in what they find, and we need to essentially develop and understand alternative techniques that takes into effect the higher order interactions in the data, such as CUNA, cumulants, redescription groups, and TDA, to get a better picture for these epidemiological questions. I just want to take a note and say that this is also pretty generic and can be transferable to any multi-omics diseases, and we do that all the time. With that, I'm going to end my talk. Thanks for giving me the opportunity to speak here. I also want to thank my co-authors and also Aldo, who had been developing the package called Matilda, which is like a one-stop shop for TDA in biological data. So please feel free to check it out. There's also Genophores-D, which is another repository that we develop in IBM Research in the computational genomics team, where we have like host of tools that you could use for your own purposes, including CUNA that I just showed, and also the paper is in bioinformatics as part of the ISMB proceedings. So feel free to check it out and give us feedback. So thank you.
So we have time for a couple of questions. Anyone? So I just have a quick one. So there's a lot of social determinants of health. So I don't know, how can you leverage that information into this analysis to take it into account?
Definitely, yeah. We did think of that earlier, but then we focused on just the demographic variables and not looking at the socioeconomic conditions, like other things. But we, for example, in UK Biobank, we study the Townsend Deprivation Index a lot as one of the social determinants of health, and we probe the same stuff. So yeah, you could. These are like external variables that you could definitely incorporate in your study and do similar analysis to find out how they interact with these comorbidities and exacerbate them or make them better.
Thank
Thanks.
you.